Okay, so welcome to this next video in the playlist on lipid modification of proteins. In this video, what we're going to talk about is the palmitoylation of IP3 receptors. Okay, so we're going to start off by discussing the IP3 receptors and the structure of an IP3 receptor. We'll then discuss palmitoylation of proteins and S-palmitoylation. We'll then discuss a complex of proteins that is present within the ER membrane known as the DHHC6 CELC complex, which is responsible for palmitoylating the IP3 receptors, and we'll see that it specifically palmitoylates, well, at the moment what is known is that it palmitoylates the IP3 receptor 1 subunit, but currently it's not known whether it palmitoylates the other two, and then finally we'll discuss the significance of this palmitoylation. Okay, right, so palmitoylation of IP3 inositol 1,4,5 trisphosphate receptors, which are often just abbreviated to IP3Rs. Okay, right. So I'll just underline and then we'll begin by discussing the IP3 receptors. Okay, so the IP3 receptors are a complex of 4 proteins which are sitting within the membrane of the ER. So let's just draw a picture of our cell. So here's a cell, and here is the nucleus of the cell, and one of the big intracellular organelles is an organelle known as the endoplasmic reticulum, which is membrane bound. Now, among many other functions, one of the functions of the endoplasmic reticulum is that it is an intracellular calcium store. Okay, so it actually has a much higher level of calcium within its lumen than is present within the cytoplasm of the cell. Okay, so calcium level within the cytoplasm of the cell is absolutely tiny. It's around 100 nanomolar. Okay, compared to an extracellular calcium concentration of around 1.5 millimolar. That's a 15,000-fold gradient. Okay, so calcium level in the cell is kept ridiculously low. Okay, however, there is one store of calcium within the cell, and this is the endoplasmic reticulum. So you have lots of calcium stored within the ER, and basically, as part of many signaling cascades, what happens is that channels in the membrane of the ER open and release the calcium from the lumen of the ER into the cytoplasm, and that calcium that goes into the cytoplasm then acts as a signal basically and causes many downstream effects. Okay, so the channel that sits in the ER membrane which releases calcium from the ER into the cytoplasm, or at least one of them, there are also others, is the IP3 receptor. Okay, so there are also the ryanodine receptors, but we won't discuss those. We're interested in the IP3 receptor. Okay, so here is a picture of the IP3 receptor sitting in the lipid bilayer of the ER membrane. Now, this side is the cytoplasmic side, okay, so this is the side facing onto the cytoplasm here, okay, whilst this side of the IP3 receptor is facing into the lumen of the ER, so this is the lumen of the ER. Right, now let's have a look at the structure of these calcium channels in a bit more detail. Basically, an IP3 receptor is not just one protein. Instead, it is 4 proteins stuck together. Okay, so I'll draw this here. So we have 1, 2, 3, 4, and I'll draw a line down there to make it more authentic. Right, okay, so this is the IP3 receptor. The entire thing is called the IP3 receptor, but it's made up of 4 separate subunits. So basically, an IP3 receptor is what you would call a tetramer. It consists of 4 separate subunits. Now, what we're going to do is pull out one of these subunits here. So let me highlight it in purple. So let's pull out one of these IP3 receptor subunits and have a look at its membrane spanning topology. Okay, so I'm pulling out one of these subunits, a quarter of the IP3 receptor, and I'm going to look at what does this protein actually look like? How does it span the membrane basically? Okay, so again, let's have the lipid bilayer of the ER membrane drawn here. So this is the cytoplasmic side and this is the luminal side. Okay, now basically a single IP3 receptor subunit is a protein. It's a single protein. So here's the amino terminus of that protein. Then it has a membrane spanning alpha helix which straddles the membrane, another one, a third membrane spanning alpha helix, a fourth, a fifth. Then it tries to span the membrane, fails to do so, and it has this little loop region here, and then it finally has a sixth membrane spanning alpha helix, which then finishes with the carboxylic acid tail, basically. Okay, so this is the membrane spanning topology of a IP3 receptor subunit. Now, these are massive, massive great proteins, basically. They are around 2,750 amino acids long. So remember, a polypeptide is a polymer of amino acids, and the number of amino acids in this polymer is around 2,750. Now, to build an IP3 receptor, we are joining 4 of these proteins together. So this tetramer here is absolutely enormous, basically, and these are giants, basically. You can see them with electron microscopes. Okay, so they're quite impressive proteins. Now, let's add a little bit of complexity into this. So basically, if you were designing IP3 receptors, a very simple way to do it would be to have a single gene, which we could call the IP3 receptor gene, and basically this could make a single protein, which we could call the IP3 receptor subunit, and we would assemble 4 of these identical IP3 receptor subunits together to make an IP3 receptor. That would be the sort of perfect simple world. The reality is it's not that simple. Instead, there are 3 different genes for IP3 receptor subunits. Okay, so there are 3 genes. One of them is known as the IP3 receptor 1. Okay, so it's the IP3 receptor subunit type 1. Then you have one that's called the IP3 receptor subunit type 2. Okay, and then finally you also have a third IP3 receptor subunit known as the IP3 receptor subunit type 3. Okay, so these are 3 different genes. They all will have slightly different sequences of organic bases, which means that the protein will end up probably having a different sequence of amino acids, and indeed they do. So these 3 separate proteins then have different sequences of amino acids. However, they are similar enough that they will all have this same membrane spanning topology, and that they can all be used to make up a quarter of an IP3 receptor. Okay, so now the question remains, how do we actually assemble them into tetramers? I.e., if we've got 3 different types of subunit that we can use in a single one of these slots, is it the case that when building a tetramer, you always use 4 members of the same subunit, basically? So you use identical subunits in all 4 slots, therefore you'd make an IP3 receptor subunit type 1 tetramer, which we might call the IP3 receptor 1. We could make a tetramer out of IP3 receptor subunit 2s. We could make a tetramer out of IP3 receptor subunit type 3s. This sort of making of a tetramer where you use 4 of the identical subunits would be called a homotetramer. So homo means same. So it's called homotetramer because you're using the same subunit 4 times, basically. However, it's not that simple again. You can make the homotetramers, but you can also make what are known as heterotetramers. And when we're making heterotetramers, the rules are more complicated. Basically, so far, what has been observed is that you can find heterotetramers which contain 2 different types of subunits. So far we have not found IP3 receptors which contain all 3 different types. So basically the rules for making an IP3 receptor are, at most, you can pick 2 different subunits that you want to include. So maybe I'll pick the IP3 receptor 1 and the IP3 receptor 3. Okay, and then what you'll do is you'll take some IP3 receptor 1s, maybe I'll take 2 of these, and I'll take 2 IP3 receptor 3s, and I'll put those together to make a tetramer. But the stoichiometries aren't really characterized very well, basically, so we don't know how many of each one you pick, and then how you arrange them relative to one another. Things like that are just not known, basically. These things will emerge within years to come, but they're not known at present. What we do know, however, is that you can construct heterotetramers where you have 2 different types of IP3 receptor subunit type in your tetramer. Okay, right. So that concludes our discussion of the IP3 receptor. In the next video, what we'll move on to is discussing palmitoylation in general, and then we'll look at palmitoylation of these IP3 receptors.